This afternoon, we're going to have 3 practicum talks on issues critical to translation, talk on clinical trials and what to think about, even if your technology is several years or even longer before that. I think we're going to hear things to have on your horizon to think about as you start to evolve technologies. Then we're going to have a talk on regulatory issues, again, things to think about early in the product cycle. And then lastly, we'll hear about funding and funding strategies for commercializing technology. So to lead off this session, it's a pleasure to introduce WGiannobile. He is really a clinician scientist in the true sense of the word, in that he's had long-term NIH funding, but he's also probably done as many clinical trials as anyone in the dental school at U of M. So truly a bench to chair-side person. He's been more than a pleasure to work with in co-directing this center. He's, in addition to the many hats he wears, he's the chairman of the Periodontics and Oral Medicine Department, the editor of the Journal of Dental Research. So he has more than a full plate, but I think he'll really demonstrate his expertise in this talk on clinical trial essentials and what to look for, especially early as you start to evolve a technology. So it's my pleasure to hand the mic over to Will.
Okay, so thank you very much, Dave. And again, I would like to thank everyone from this morning's session. I really thought it was an outstanding showcase of the technologies that are being developed as a part of the Regenerative Medicine Center. And so, our charge in setting up this afternoon's practicum sessions was, as you can see, we've got some practica on clinical research, regulatory, and funding. And much of this is really designed for the basic translational researcher. The types of materials that you will see here is really looking at phase 1, phase 2 clinical trials, and then eventually what might be needed for phase 3 clinical trials and eventually getting approval. And one of the biggest aspects is indeed looking for the end in mind and then working your way back. And so when Kay Fuller and I, she'll be the next presenter, we're discussing our talk, she's going to have more of an emphasis on the preclinical requirements for bringing new technologies to the FDA. And so I'm going to start off with really the clinical side, so you can look at identifying what are these key clinical endpoints that might be surrogates of what's being done from the preclinical side. And then in addition, this regulatory landscape, we have Kay Fuller, who's working with us, Jeannie Wright, they're sitting in that middle row there, really as a wonderful resource that many of you have worked with over the past, the year as a part of the consortium. So they're a great resource to help answer questions for us. So I show these icebergs here. They're from Lake Michigan and Lake Superior. So I think it's appropriate for what we've been challenged with this week. And I'm sure some of these icebergs have grown. This is from the Pictured Rock area, and quite beautiful. So as we think about the consortium and having the support by the NIDCR, and again, looking at the charge that we have with these regenerative medicine resource centers, is really looking at how we can propel novel therapeutics from the preclinical arena to FDA submissions in human clinical trials. So a part of what I will be presenting is really trying to, again, be in strong alignment with the NIDCR goals for clinical translation. And this approach has been set up by our organizational structure to balance clinical needs with the technological needs to move these technologies forward to help transform patient care. So this is part of a white paper. Dave had mentioned this earlier this year that both of the centers had put together. And so I would encourage you to take a look at it as a reference, and really is helping underline the goals that the NIDCR had. And so as we think about regenerative medicine technologies, it's an exciting period, despite the fact that, I sit on the FDA Dental Devices Advisory Board, and I've only had to go to one meeting in 13 years as a part of the committee, and it's really a challenge to see that we've had so few PMAs come through for regenerative technologies. At the same time, the FDA, when you interact with them, they really want to see more of these coming in from the PMA route versus the 510K, and we're going to hear a little bit more from Kay about that. But there is definitely, as it relates to cellular therapies, stem cell therapies, it was mentioned earlier that there are going to be more and more submissions to the FDA. And so some of the guidance that is being directed by the FDA is looking at, how are these new therapies going to be evaluated? And it's a challenge. Related to this, and it was brought up earlier in terms of the new clinical studies, this consortium and a discussion we had with Lillian Shum is that, next year, I would stay tuned because we're looking to have a workshop that's going to identify many of these endpoints, combining preclinical studies and clinical studies for the regenerative medicine space, because it's really uncharted territory for some of these new biologics and cellular therapies. There's just really only a handful of these technologies that have been out there. So really want to encourage their development. So you've seen this slide before in terms of how our center is set up. And one of the aspects as a part of this presentation that I wanted to highlight is that this is really focused on the preclinical development to get the new technologies into the clinical arena at the doorstep of the Food and Drug Administration for that delivery. And so these three presentations this afternoon will focus on how can we get the exiting of the projects to clinical trials, establishing the licensing and development to go into early phase clinical trials or a 510K type of route. So that's one of our ultimate goals as a part of this center. And many of the challenges that we've seen is really this conversion from the outstanding basic science research and translational research, but at the end of the day, how many therapies are actually benefiting patients. So we're not just trying to do an interesting phase 1, phase 2 clinical trial that never goes anywhere. And as a part of this, to get it into the clinic, so the chair side in the case of most of the dental technologies, we're seeing that we're either looking at a commercialization pathway or one that might be for orphan types of diseases, conditions, where it might be a hospital-based technology that can advance patient care. And so this is something that continues to be a challenge both for medicine and for dentistry. And many of you have seen similar types of timetables on the development of new devices and biologics and drugs for clinical application. And when you look at what is truly involved from the initial discovery phase and preclinical testing, and then some of the steps, and I'm going to go through these in more general terms, from submitting an investigational new drug application all the way to doing phase 1, 2, 3 human clinical trials that would then result in a technology that could go into the clinical arena. And so as you think about that discovery phase and wrapping up the intellectual property and licensing, you can see how much time could be eaten up when you have this patent life of approximately 20 years and making sure that that intellectual property is well wrapped up. And so that's been one of the aspects of our resource center, working with investigators to make the most of this timetable so they can enter in to the clinical trial phase. So it is a long process that's involved, and I think it's something that also the FDA and in Europe, the European Medicines Agency, they have recognized this and looking at ways to try to accelerate it. And I know we have several people in the audience who have done taking these technologies to the clinic. And it's a challenge. And I don't think anyone has an easy answer on the best way to get these technologies into the clinic. But I think as a part of the center, trying to work with our investigators to propel these technologies. So with the regulatory process at the investigator level, so many of you working at in your universities, and some of your work in the corporate world, you know, it's almost like the principal investigator has this target on his or her back, and they have so many roles and responsibilities in terms of you know, compliance and regulation that, you know, it really requires a team approach. As you look at many of these different, you know, putting in an IND or an IDE, GLP practices, the applications, biologics, financial disclosures. So there's so many areas of expertise that are required for doing human clinical trial research. One of the things that you've seen, and we've tried to, you know, when working with investigators, is taking advantage of that to assist the investigator in moving these approaches forward. So the significant responsibilities. At the University of Michigan, we have an investigational group that works with clinical trials, and the average clinical investigator is responsible to 9 different entities at the university. So, you know, it could be Institutional Review Board, the Office of Human Research Compliance Review, their local IRB, so many different entities. So it's a lot for an individual investigator. And so as you get started in developing clinical trial research, it's important to deal with all of these different aspects. And in terms of these quality standards, this is going to be an aspect that I'm going to have Kay Fuller go into a little bit more detail. But if you look at the ethical principle standards and what's going to be involved in eventually putting in an IRB, Institutional Review Board application, following those various guidelines from ethical principle standards. And as again, you think about preclinical research, things that you can do preclinically and then clinically comparing to a standard of care, there are many ethical aspects that are not going to allow you to do the quote unquote ideal experiment because the welfare of the patient is critically important. And then we even heard a little bit in terms of quality standards, ISOs, that are required by the Food and Drug Administration for different, you know, it could be a biologic biomaterials that are going to be utilized as a part of the clinical studies. So this aspect is important to have in mind. And so in the preparation of an application for the FDA, these aspects would having good clinical practice guidelines for either medical devices or drugs and biologics. This is, again, working with your regulatory teams to help support you in those applications. And then there are the national regulations on clinical trials, ranging from non-interventional studies, which, and I'll define this a little bit later, these are not typically studies that are going to be going through this quality standard aspect versus the phases of human clinical trials for a drug or biologic, or a pilot/pivotal study for a device. And we'll talk a little bit more about that. And then this retrospective. So on both ends of the spectrum here, those types of studies are really not subject to the FDA types of standards. One of the things I'm just showing you from the textbook, there are 3 textbooks that our team, and actually many of the people in this room are co-authors of these books that we're making freely available for everyone here. So you'll see on the last slide that those materials are there for your use. All right, so as we look at the design of a human clinical trial and identifying, you know, is this interventional? Is this an interventional type of study, or one that is not an experimental study? These are some of the key aspects of discriminating between an interventional study or having an observational study. And it's really the interventional study that is going to be subject to those aspects that one would need to go through the FDA And so the basic questions here in an observational study: is there a comparison group? And if it's not, it's more of a descriptive study or an analytical study that could go into, and we're seeing, you see much of this in dentistry, a case report, case control, case series types of studies. And we see these either at very early stages as off-label, or this could be, and we talk about a bit later, in phase 4 post-market approval. And so something that's already approved, maybe for some other indication in selected cases, versus the experimental study looking at randomization, this being a key aspect in doing a randomized controlled trial for the therapy versus the clinician guiding a therapy for a specific indication. So this, sometimes a non-randomized controlled trial is still viewed as very valuable. Maybe it's based on a specific treatment protocol. So maybe you're doing a bone regenerative device in the posterior mandible, and that severe defects are going to receive one therapy just as a standard of care versus a less severe defect, and there's a protocol, versus a randomized trial where you're really truly randomizing for a different therapeutic for that patient. Okay, so this particular slide I'm going to go through, this is a process in terms of considering doing a clinical study as a stepwise process, and looking at the input in terms of the study activities on the top, and then the output on the deliverables. And this was part of a work that Kay Fuller and Darnell Kegler we did together, looking at some of those key aspects that you can use as a reference in the development of a clinical study, and considering all of the pre-study activities in the development, identifying the patient populations, the study design that's going to be required. I mean, this morning we were just talking about that. What would that eventual study look like? What do you see as that design, and what type of defect might be being treated? And then you can see the assembly of all of the key individuals in your team, biostatistics from the research grant side, and then eventually from the FDA. And then you've got the study approved activities ongoing. And so again, so many aspects that are critically important for the development of that study that could eventually then lead to an approved study that has high quality data going through the appropriate institute at the FDA that could then lead to a new drug, dental biologics, or device. So some of these basic phases of human clinical trials, and again, one of the goals of this consortium is getting a new technology into a phase 1 human clinical trial, or as we'll talk about a little bit later, a phase 1, phase 2 clinical trial. And so phase 1 trials are typically a purely safety type of study, and the primary goal of these is to assess, safety, and oftentimes done in a group of healthy volunteers to see, it might be used eventually for assessing toxicity and some initial dosing information. So in terms of the phase 2 studies, these oftentimes are one or more, so it might be a 1 or 2 or 3 center study that's looking at some preliminary efficacy or dosing requirements and preliminary evidence for efficacy and supplementary data. So this is where, in the phase 2 study, and very commonly phase 2 and phase 1 are combined, especially for some drug or biologic that has already been tested in humans, where one is these types of studies you can probably have a much better control of, because you have all the centers there. You might be able to really identify, let's say, for example, the defect topography that you might be looking to repair, and seeing which types of cases will be best suited to your regenerative device. Some information on dosing to see, as we go from an animal model to the human, there's always there's quite a learning curve in trying to find that sweet spot of the drug dosing that might be most important. So a lot of activity can be worked out in the phase 1, phase 2 study. You may not have what seems like the blockbuster drug at that point, but you're going to be able to see how those patients are responding and some of those early clinical evidence. And then the phase 3 study, this is, as you think about the costs and development, this is where it really goes beyond what any academic could do. The phase 3 studies are usually large, so in the hundreds or thousands of patients, typically multi-center. And in dentistry, we've seen where these multi-center studies might be a dozen centers to, 20, 30 centers that might be involved in testing the effectiveness of a new drug. So this is looking at, again, all of these are examining safety, but effectiveness, looking at the generalizability of this particular technology. Again, you're not going to be able to control things as well as what was done in a phase 2, because this was either a single or a couple of center study, but now you're putting this into the hands of, it could be, dozens of clinicians, where there needs to be training, calibration, trying to control that as much as possible, but you're looking at, the diversity of, it could be the racial, ethnic profile of the patient's age, geographic areas, and so you're conducting that study in a way that it's a more generalizable result. And then in the phase 4, these are the post-market studies. So FDA approval to go into the marketplace occurs after phase 3. And then at phase 4, now you've gone from maybe hundreds or a few thousand patients have been treated. Then there can be MedWatch reporting and events that could be identified, and it might be tens of thousands or millions of patients. And this is very informative to the FDA. These are where sometimes we start seeing approved indications that clinicians are utilizing. I think, I don't know how many of you know, what is the percentage of off-label use of regenerative devices? Does someone want to loft out a number? Okay, you're conservative. It's higher. It's approaching close to 80% of off-label indications that you'll see in the clinical arena, and that's where oftentimes there can be problems. But that's where oftentimes clinicians are starting to see there might be another application, but it's an off-label application. So that's why, as we see, almost the Wild West of some surgeons in terms of different technologies and how they use. Yeah, that's obviously going to be highly variable depending on the study design. So many of the studies in dentistry are 6-month to 12-month studies for the approval of a regenerative device. And I know some people here have looked at those. So usually in a 6 to 12-month time frame. And I've seen, depending on, let's say you're using a local drug delivery, not combined with a surgical treatment, it's going to depend on the numbers and types of procedures, the numbers of visits that a patient will need to undergo. So some of these studies might have a cost per patient of, as low as $1,000 a patient, to many others that are $10,000, $20,000, $30,000 a patient. In general, within dentistry, I don't know if there's anyone else here who has had some experience. Probably not much more than that in dentistry. But we have some people in the craniofacial area. I'm sure it would get much more expensive on a per patient basis if someone is trying to do a large cleft defect or large craniofacial defect. So that could, right. Right. Yes.
Sometimes some of those procedures can be actually charged to the insurance company, depending on what it is. So when you set up some of these things, you need to set up a billing schedule. And some of that could be investigational, and yet some could be charged to the insurance company, depending on what it is. It varies from situation to situation, obviously. So that can mitigate some of the costs.
Right, yeah, and Steve's bringing up a good point, especially in medicine, right? These procedures are billable procedures. because you're using a standard of care comparator. Our challenge in in dentistry is that many of these procedures may not be covered, or they're partially covered. And in general, I've found in dentistry, and I know we've got several people from the insurance industry here, it becomes a challenge to have much of that, especially if it's an experimental approach. But you're absolutely right, and I know the Clinical Trial Center here at U of M, you look to reduce those costs to the patient and the study as much as possible. Yeah. Okay, I put this graphic up, and this is one that Kay is going to get into a little bit more, but I'm showing the value of this particular graphic is showing this connection between what we're going to hear about our discovery research, the benchtop research, preclinical in vivo investigation, and then how that's going to interface with doing the human clinical trials. And so much of our wheelhouse as a part of this center is really focused on this preclinical development. Many of these studies done under GLP or GMP conditions that would then eventually be used for a submission to the FDA. And then once you get through that, and I've even heard we've got some people here that are at this 30-day review to do the human clinical study, which is exciting to hear, then a determination is made, okay, can you do the human clinical trial based off of these preclinical studies? And so we've had a really, I think, a good iterative process with the investigators and working to guide them, because you don't want to be doing large preclinical studies and then the FDA is going to come back and say, we don't like that study design. We don't think that that's a good endpoint. That might have been valuable for you to do some of the discovery research to see, does this technology work? But you don't want to do a study that you think is going to be setting you up for the FDA without getting their iterative feedback that, yes, this is a good study design, that it eventually would get you into the human clinical. And I know we've got people with some experience with this. Yep.
Sorry, so your green box there is where many of us are with this program. And, it's hard to know whether you actually need GLP for some aspects of your preclinical work. I mean, I think particularly doing GLP in large animal models is very challenging. And I don't know if there's anyone who can comment on this. And when the FDA sort of says non-GLP is okay.
Yeah, that's a good question. I think I'm going to let Kay. I think she's going to spend a fair amount of time on that. It's an excellent question, because many people assume they have to do GLP to go to the FDA, and as you said, it's not in all cases. And so you want to know that, do you need to do that? For example, our animal facility here does many things GLP-like, but we don't have a GLP facility for our preclinical animal models. I believe at Pittsburgh you do have a GLP facility. Charles, you have a GLP facility that sometimes, yeah. It's GLP-like, but some of these CROs for preclinical testing, oftentimes they will do that, but it really depends. And I'm going to defer that a little bit more to Kay out of the interest of time, so I'll have her come back to that question. Okay, so as you look at these quality metrics and what's required to go to the FDA, if we look at for IDEs, or investigational device exemption, so this is for, in general, some of the biomaterials that are being evaluated as a part of this consortium, really going from this prototype development and bringing this towards the human clinical trial. So again, it's the pilot study followed by the pivotal study that will lead to the FDA PMA. Okay, so what is a clinical trial? And just some of the basic, and I'm going to go through some of this material fairly quickly out of the interest of time, but I touched on this earlier in terms of a prospective study comparing the effect in a randomized way of an intervention in humans. And again, as I mentioned, it's not an idealized experiment because we're looking at humans. The standard of care always has to be incorporated as a control, and the standard of care keeps, raising up, and in terms of what we can do for patients to do those particular types of studies. The classic example is you can't do a randomized controlled trial on parachutes, on whether they work or not. So you can't use that standard control. All right, so in terms of drugs and devices, it's the starting point, the most typical type of study, again, just for a summary of the phase 1, primarily safety study on human pharmacology. These are some aspects of the study looking at the safety, pharmacokinetics, drug metabolism, looking at a new drug and all those types of effects in the normal healthy volunteers versus the therapeutic study in the phase 2s that are really looking at this laundry list of aspects in terms of looking at dose escalation, preliminary efficacy. And then for medical devices, so instead of being called phase 1, 2, 3, are called exploratory or pilot and confirmatory. But the exploratory is very similar as what you see for a medical device or a new material, that it's very much like a pilot, showing the proof of concept of that technology to, for example, repair a defect and demonstrating its safety. Whereas the confirmatory study or pivotal study is really looking at, in this large patient population, multi-center investigation to promote in a controlled environment, oftentimes sponsor-initiated for that approval. Choosing the experimental design, there are many different characteristics, and we're not going to be able to go through all of them, but these are many of the different considerations that are involved, from the ethical considerations, the previous information from the preclinical animal studies. And, in terms of those different phases of studies, from phase 1, looking at, how much toxicity is one willing to accept. So when we look at dental, we're looking at non-life-threatening diseases, there's going to be very little toxicity that can be accepted, versus if you're looking at a new cancer therapeutic, you're going to be able to accept more toxicity. And so this is, again, one of the challenges within dentistry, looking at, it has to be highly safe, minimal toxicity, and, will you be able to hit this maximum tolerated dose, looking at some of that dosing in the phase 1 study. And again, phase 1, looking at some of these studies. This comes from a chapter from Sam and his team, and looking at different disease types, combinations, or single agents, and that study design that would be done for a phase 1 study, and examining, what is this therapeutic window, having that optimal dose and trying to stay out of the area of toxicity and having a minimal amount of efficacy. Oftentimes, you're not going to be able to see it in the phase 1 study so well, but you are going to be maybe doing more of a dose escalation as you enter into the phase 2, but hopefully you're going to have a good idea of that therapeutic window from the phase 1. Phase 2 study design, and this is the point where oftentimes decisions are made whether or not a company should invest in expanding this technology further. So then the technology might be killed at this point in terms of seeing this initial phase 2 study design. It helps to screen out ineffective drugs. So if you just cannot see an effect in a human, like what you were able to see in the preclinical animal model, what is that? And then you might also, on the flip side, find these more promising drugs for further evaluation. And then, again, further refinement of the safety and toxicity, because now you're maybe having a more standardized type of defect that you're treating for clinical application. And so this gives just a little bit more detail. I've touched on this already in terms of the patient population size, some of the questions that are asked, endpoints again, and we're starting to see more and more endpoints that are non-invasive. So we saw earlier Stephen Blumenbergen, who had showed coupling a diagnostic, it might be a biomarker assessment, with the clinical outcome using non-invasive imaging technology. And the FDA is, they are very open to hearing about these other new diagnostics that can be coupled to identify if there is indeed a clinical effect. Okay, and then this touches on historical controls and the use of a standard of care therapy. So it could be a regenerative agent where there's surgery alone and a bone replacement graft versus using your regenerative agent in combination with that. And so how much efficacy is there already there? It's going to be very difficult to see that response. And what the FDA is also very strict on is for all of these studies, using as few patients as possible. And we'll hear more about biostatistics later, but you don't want to overpower these studies. Enrolling 1,000 patients to show a less than a 1% increase in regeneration. That's just, it's not going to be appropriate and won't be clinically relevant. Okay, and I'm going to move through this. So as we start to look at, again, the end in mind and moving towards putting these into pivotal studies, this is just an example of as you start to move towards phase 3 or pivotal clinical trials, to give yourself an idea of what is really going to be required in developing the multi-center study, having a data coordinating center, having a data and safety monitoring board. So this is a group that's going to help identify, should you stop this study? Is there a medical question? How do we deal with adverse events that are occurring, especially with multiple centers? Oftentimes there's a single institutional review board that's looking at all of the patients. This is more ideal than having 10 universities with 10 IRBs assessing what's going on. This is a nightmare, and I think many of us have experienced this. Very difficult in standardizing that. So there's so many different elements on the outcome measures that are assessed, the different investigators, and the coordination. So having these coordinating centers working with the PI and medical director is very important. And Kay is going to touch on this a bit later, but I think as we start to identify, many of us, when we do use a regenerative agent, we want to show it's the best, it's superior. And that's a game changer type of technology. However, looking at equivalence or non-inferiority, if you have a technology that is not significantly different than something that's already on the market, but it might have less morbidity. And so the numbers of patients that might be able to receive that therapy might be much different. So this slide here shows what one would expect in terms of something that's truly superior. And this is all has to be done with the biostatistician, that it's much better than the control treatments. Equivalent is really the result, and statistically is very difficult to demonstrate, where you truly have this overlap of treatment groups and control groups, and they have so much overlap, they're really equivalent. And then the non-inferiority, this is where oftentimes with the 510Ks, it's an easier bar to demonstrate. This might be better, but you can't prove superiority, but you can also say, well, it's not worse. And it doesn't sound like a great endpoint to have, but we see this is what, through the 510K process, is probably the most common indication. Okay, and so the analytical plans depend on the study goals. I'll just leave this here for a reference because we could have a whole session on the statistical aspects that are involved, but there are different statistics, obviously, as you move up from phase 1, where these are descriptive studies, pilot investigations, where you're just trying to identify what is the effect, and then the head-to-head comparison at the phase 3 requires a very robust statistical analysis. So my last couple of slides before I stop is, just really looking at what is going to be required with your study team as you develop your clinical trial. And again, it's a major team effort where you might have, I showed that one slide of a multi-center phase 3 trial. It's really set up with the chief investigator. This might be the person who has really come up with the idea for the new technology. It may not be involved with seeing patients at all, but then could work with multiple principal investigators who are in charge of each study center. And then we have study managers and study coordinators. These are some of the, I would say, the most important people in the clinical study team to maintain the integrity of the study in terms of how how it can be well organized. And I'm going to talk a bit more about study monitoring, but it's really working collectively with the study coordinators and those who've been involved in phase 3 clinical trials. You'll be able to tell right away if this is a good center based on the quality of the study coordinators oftentimes, because then everything will flow from them in terms of there might be the treatment clinicians, the surgeons who are delivering the regenerative device, the examiners who are separated from those surgeons to look at the outcomes so they're not biased, and then the support team that's involved, other clinicians, recruitment, this being a critical piece. So it's a very strong team approach that's required for the success. And so as one looks at, there's this crude ratio of a 3-to-1 in terms of clinical visits, thinking about how many patients sometimes you need to see before you can identify a good patient that could fulfill inclusion and exclusion criteria. And those of you involved in clinical trials, this patient recruitment aspect is the make or break, right? And some centers cannot recruit well. They might be very good in identifying inclusion, exclusion criteria, good data quality, but if it takes them 2 years to recruit for a particular study, that's just too long oftentimes based on the timetables that are put forth. So it's really broken up into these 3 parts of the setup of the study, working on getting all of the approvals from the IRBs, finalizing all that coordination with the FDA, identifying the study team, and this upfront part is critically important. And then things will usually fall in place after that in terms of those clinical study visits. The question here in terms of, what does it cost per patient? It's really going to be defined quite a bit on this aspect and how often these patients need to be seen. And then for a patient to complete and exit the study, you don't have a high dropout rate, and then to eventually be able to report the results, publish them, and submit for FDA approval. Clinical trial monitoring. So this is a part in our MyApp program, which actually Kay helped develop here, that oftentimes the monitoring and following these patients up, maintaining the quality of the data, having that evaluated by an outside non-study team member is very important. So for an external data and safety monitoring board, oftentimes they're looking at the data. There might be an identification to say nothing in this study seems like it's working, and there could be certain rules to stop the study, either due to no efficacy or there could be major safety concerns. And so this is an external body that is going to help design this. And so NIDCR has organized these data and safety monitoring boards, and it's usually a very rigorous process to maintain the quality of the study and identify those studies that should continue, those studies that maybe should be shut down, and those aspects. So, considering using clinical research organizations to assist, NIDCR has their own group that they've worked with that it's external to the NIDCR, and most individuals, once you get into phase 1, phase 2, should be considering the use of CROs to people who have expertise doing this time and time again, not using the clinical investigator. And some of the questions in terms of patient safety versus study integrity, the early stopping rules, these are all aspects, again, that the Data and Safety Monitoring Board, and then some of the, what the clinicians are seeing, the examiners are seeing, and, would there be an interim analysis done? Should we, peek at the data? If that's going to be done, that has to be approved by the IRB and the FDA, and the different regulatory groups are sometimes using the Data and Safety Monitoring Board. Okay, so as I wrap up in the next couple of slides, Dave showed this slide earlier this morning, as a part of our center all of these different aspects in the conduct and the development of new technologies that will be translated and getting into clinical translation. And so it's really important, and we were just talking at lunch, I was chatting with Jeannie Ambruster and Francine Berkey, and looking at how this should not be siloed. It really is, again, looking at the end in mind and how you can really work together to put all of these elements in a quite a combined way to synergize what you're going to be able to accomplish in the study. And so sometimes you might be focused so much on manufacturing, but then at the end, this isn't a strong enough clinical need. You're not going to be able to develop a clinical study that will eventually show that this particular device works, and it's really not a good use of resources. And again, just reiterating, using these different resources that we have as a part of the center, here and available to assist you with the development of your clinical studies. And very important that we look at how this technology will look, how the clinical trial will look. It's very important. It's not something you just put off to consider later. It has to be considered up front, especially if you're looking at, you've got licensing, you're looking at corporate partners, and you have to know what this technology is going to look like. And so we have our support groups there. And then here we have just some of the resources. These are all freely available, and acknowledging some of the materials shared by Dan Clow, who helped run the Research Development Corps here at Michigan, and others. And then again, this slide was shown earlier, and again, it really takes a combination approach, working with the many stakeholders that we have here as a part of the consortium, and working with NIDCR support to help advance clinical research. So with that, I think maybe have a couple of minutes for a few questions, and thank you. Yeah. Sure. Yeah. So Matsumi has made all these slides available, and we've also made all these presentations will be part of a webcast. So I think the video recordings are also, or the podcasts, those will be available as well. Yep. Steve?
Based therapy, it's a whole different world. Dosage, potency, toxicity, pharmacological approach is much more complex and involved. Just as an FYI for people pursuing that.
Right. Yeah. So what Steve's saying is it's even much worse than what I've just shown in terms of many of these technologies. And Steve would know, he's a great resource if it comes to cell therapies. He's still, he's still with us, after all he's been through with that. But it is a big challenge, and that was when I showed that snapshot from that New England Journal paper, and this stem cell consortium that's been working to help move these technologies forward, because the regulatory path, as you've said, is it's a high hurdle for cellular therapies, and that's where I think as we look at the prioritizing and de-risking technologies, right? This is difficult with both the autologous or the allogeneic cell stocks that are used. You could have a whole session on that. Lillian. Yes. So I've got that one shout out to Mishar that runs our CTSA. Pittsburgh has one as well, and absolutely these resources. So this is Clinical and Translational Science Awards, or CTSIs, the institutes, and these are institutional NIH supported groups that help accelerate and propel clinical studies. And so there's so much infrastructure. So Jeannie Wright, who's part of our MyApp program, that's really a component of Mishar, and there's so much expertise at the university level. So either your local university, or we've tapped in as a part of our consortium to use a lot of different resources. So an example is I mentioned Dan Clow and the Research Development Corps. That particular group, we work with investigators on that study launch, and that's using their core and many of the people from biostatistical support in identification, the planning of your preclinical study. These are people who are all part of our CTSA. So absolutely, this is a huge resource that has really propelled clinical research at universities. Okay, so I just saw the sign that my time is up, and so I guess we'll move on to the next.